Okay, this is Unit 2, Cells and Biochemistry, and this is Lesson 3. This lesson is just going to review pH and talk about enzymes. So we've been doing some labs with indicators, and a lot of the indicators that we've been using change color in the presence of a certain substance, like a lipid or a carbohydrate, or we've been using the litmus paper, and you're detecting whether something is an acid or a base. So that's directly related to the pH scale, where pH is a measure of the acidity or alkalinity present. And what it really measures is the relative amount of what's something that we call hydronium ions, which is H3O plus, and that plus means it's an ion, and these are what's called hydronium ions. So basically it's measuring the amount of H present in a solution, and anytime we have H in front of a compound, it's an acid. So hydrochloric acid is HCl, nitric acid is HNO3. And in one part of the lab you were measuring carbonic acid, which is what happens when you exhale carbon dioxide into water, which caused the indicator to turn yellow, and that's H2CO3. That one's carbonic acid, and that one was directly related to the lab. Okay, so as you know, the pH scale ranges from 1 to 14. If the pH is 1 to 6, it's an acid. And if it's 8 to 14, it's a base. What you might not know is the closer it is to 1. So here, closer to 1 is stronger. So if I have a pH of 1, I'm a stronger acid than I am if I'm a pH of 6. And here, closer to 14 is stronger. And if I am right in the middle, 7 equals neutral. So one thing that we know for sure is usually neutral is water, H2O, and that's important. Okay, pH is important because it directly affects reaction rates. By changing the shape of enzymes, remember enzymes are proteins, and preventing them from working efficiently. So if your pH is too high, it'll change the shape of an enzyme and it won't work effectively. So let's talk a little bit about enzymes. They are biological catalysts. So the definition of a catalyst is something that speeds up the rate, how fast it goes, of biological reactions. They are proteins, as I just reminded you, and they have a very complex three-dimensional shape. The thing the enzyme works on is called the substrate. So here's my enzyme, here's my substrate, and the shapes must match. It's like a puzzle piece. Once the enzyme and the substrate link up, it's called an enzyme-substrate complex. So when the two pieces of the puzzle are put together, that's what's formed. afterwards, the products no longer fit the active site. So once the enzyme and the substrate hook up and they do what they need to do, the products that are released now have a different shape and they won't fit back into the active site. This is referred to as the lock and key theory. Or one key only fits one lock. Only one substrate fits one enzyme. Now, one thing that's cool about a catalyst is that it's never used up. So this enzyme that is a protein, that is a catalyst, is never used up. Therefore, it's available to catalyze further reactions. Okay, so let's review now. Enzymes are catalysts that control the rate, and in parentheses you can put speed, of chemical reactions. Now, a chemical reaction can involve synthesis, and we learned what synthesis is. It's when two or more simple compounds combine to form a more complex one. So here you have this red circle, compound A, and compound B moving into the active site. So this is your enzyme. These are your substrates moving in. This is your enzyme substrate complex. This is where the work is taking place. And then you can see that A and B were joined together and the new product is released. And then the enzyme is ready to go back and do the same thing over again with a new A and B. Now this can also happen in digestion. Digestion, remember, is the breakdown of food into smaller usable substances that can be easily absorbed by the body. So here you have your enzyme, and your substrate moves in, forms the enzyme substrate complex, and it breaks down this big piece into two smaller pieces that can be used by the body. So synthesis is putting things together. Digestion, okay, is another word that we call, is an example of decomposition. You're decomposing. So digestion is an example of decomposition, breaking something down. Enzymes will only work if they match the shape of the substrate they are working on. Enzymes are like puzzle pieces or a lock and key with only one key to open it. They are specific to one substrate. This is extremely important. So let's look what happens in some examples of digestion. When you have digestion in your mouth, the saliva also contains amylase. And we're going to write this on our notes. I want you to write it under your digestion. Usually anything that ends in ASE ending, indicates it's an enzyme. So that's your clue. And if you know that what enzymes do, then you don't even need to know the word. And it digests starch into maltose, which is a type of carbohydrate. Digestion in the stomach. The stomach wall produces pepsin. This is a different type of enzyme. This one doesn't end in ASE, which digests protein into amino acids. And then the pancreas also injects a range of enzymes to complete digestion. The biggest thing that I want you to know about the products of digestion is that carbohydrates are digested into sugars. And we learned that. Your building blocks of carbohydrates are simple sugars, monosaccharides. Monosaccharides are put together to make disaccharides and polysaccharides. And the products of digestion of fats, you also learned, are glycerol and 3 fatty acids. Okay, let's go back to our notes. 2 things that can affect the shape of an enzyme and make it work less efficiently include temperature and pH, which we already mentioned. An out-of-shape enzyme that doesn't work because the substrate doesn't fit is called denatured. And let's take a look at a couple of those. So the enzyme reactions rely on successful collisions between the enzyme and the substrate. So the enzyme and the substrate have to collide, they have to be a perfect fit, and they hook up. When you have an increased temperature, this increases what's called kinetic energy. Kinetic energy is energy of motion, so the pieces are moving faster. Beyond the optimum temperature, usually around 40 degrees, there's a loss of 3-degree structure of the enzyme. So the enzyme starts changing shape once you get above 40 degrees. It's now denatured. There is a loss of specificity, and therefore it will not fit with the substrate and it cannot function and do its job. The other thing that affects the enzyme is pH. Remember, I told you pH is when you have hydrogen ions present, or hydronium. And the pH is too high. It again causes the enzyme to change shape and denature it. So back on your note sheet, I want you under temperature, okay, to write over 40 degrees Celsius, enzyme is denatured. And the optimum pH is best around 7, which remember is neutral. There could be some that work better a little bit lower, depends on the actual enzyme, and we'll learn about a couple of them in the stomach that work best at a couple of different pHs. Okay, that's it for the lesson.